Everyone. Hello, everyone from wherever you are. Welcome to this session. It's a second part of our two-part program on the next generation of cell biology. It's honoring, today we're honoring, well, the whole program is designed to honor exciting science that's ongoing and published in MBoC. And in particular, today, we're fortunate to begin with a keynote presentation from Dr. Xuebiao Yao of the University of Science and Technology of China. And then after Xuebiao's talk, we'll shift focus to the next generation of scientists with 6 lightning talks from the recipients of the MBoC Early Career Award. I'll talk a little bit about that early career award when we get to those presentations. But first, let me introduce Xuebiao. He earned his PhD at UC Berkeley under the mentorship of the late John Forte and went on to complete an American Cancer Society postdoctoral fellowship with former ASC president, Dr. Don Cleveland. He launched his independent career as a tenure-track assistant professor at the University of Wisconsin, and later received a Georgia Cancer Coalition Distinguished Scientist Award, continuing his research at Morehouse School of Medicine and the Winship Institute. In more recent years, Xuebiao has been a driving force in building and inspiring China's cell biology community. He helped establish the Keck Center for Organoid Plasticity in 2017, and in 2019 became the founding director of the Ministry of Education Key Laboratory for membranous organelles and cellular dynamics, the first of its kind devoted to studying how molecular machinery governs cell quality control and renewal in live organoids. I met Xuebiao at last year's ASCB meeting and was struck immediately not only by his scientific vision, but his energy and his warmth and generosity. He's a towering presence in Chinese cell biology and a true friend of both American Society for Cell Biology and molecular biology of the cell. So Xuebiao will be presenting, I can't quite read it on my screen just yet, on biomolecular condensates and how they guide chromosomes and dynamics in mitosis. He'll have a 13-minute or so presentation followed by a 5-minute question and answer. And then we'll move on to the, after that, we'll move on to the early career lightning talks. So Xuebiao, welcome.
Thank you, John, for your kind introduction. And let me try to share my slides. And I think it's a really wonderful opportunity to see all the young speakers and also I think colleagues on the Zoom. And this actually probably spent the continent of American and also Asian Pacific region. So can you hear me all right? Yeah.
Yes, we can.
All right. So I think, well, this opportunity brings me back to my graduate year in UC Berkeley. So actually the cover is really the MBoC paper by which we collaborated with Dr. Jennifer Lipcomb-Schwartz using this super resolution. So the reason get me into the cell biology was quite simple. That's actually in the 80s. Some of our speakers today may not be on Earth at that time. I think it was the Helicobacter pylori, which is causing gastric ulcer and also gastric cancer. So the reason for me to study in UC Berkeley is really looking into the cellular mechanism. So my very first publication is really identify actin isoform in gastric choroidal cell, and this is the cellular unit which involved in digesting protein and sterilizing the bacteria in the stomach. And I was very curious because 2 actin isoform beta and gamma, they only differ by 10 amino acid, but at the cellular level, they really, one is localized in the apical membrane and versus basolateral. And the key player of this organization is a protein called azrin. And during my study, we realized actually there are mitotic regulators such as BABA1 and really often mutated in gastric cancer. And there was a quantitative study a few years ago. In fact, in adult human, every second, roughly 3.8 million cell division. So that became obviously cell division control is very important. So I actually studied cell division in Don Cleveland's lab by focusing on the very important structure called centromere, which is situating in the constricted region of centromere. I think Amy is going to talk about the centromere as well. And this is a device by which spindle microtubule attached to the chromosome drive its movements during cell division. And when I first started build this cellular dynamics unit in China as a young professor, and that was about the time when the human genome was roughly completed, meaning the human genome turned out to be relatively simple compared to the initial prediction. And of course, at that time, we thought about most of those post-translational modification and also single nucleotide polymorphism may play in role for the micromolecule function in space and time. So the purpose of this unit is really look at the molecular machinery and also organelles in quantitative manner at the mesoscale, meaning looking into the nanometer scale and micrometer scale. And that's actually the objectives for our program. So we started using proteomic approach, really looking into the identification of human centromere proteins and try to ask a question, what is the molecular composition of human centromere? And given the conservation of centromere organization from a single eukaryotic cell, such as budding yeast, to human cells, but there are some other evolution going on. So we started using a molecule called SAMPE, and I think this is going to be a fashion topic because later on, I think you will hear the SAMPE function in different contexts in the neuronal cell. So we basically, this is actually the mitotic kinesin first discovered by Tim Yan and Don Cleveland. Tim was Don Cleveland's postdoc at that time, and then he was a faculty member in Fox Chase Cancer Institute. So SAMPE is a very long protein, roughly 2,700 amino acids. So when you stretch out, it's roughly 300 nanometer. But this protein actually has a cell cycle regulated fashion, and we use this as a molecular handle to isolate that the protein binds to SAMPE at the different stage of mitosis, because this mitotic kinesin associated with centromere upon the nuclear envelope broken down, and also SAMPE degraded by ubiquination mediated process at the end of mitosis. So we actually, surprisingly, we actually identify a different post-translational modification and also new components. So one of the post-translational modification we identify is actually Aurora B acetylation. So this is actually using biochemical characterization. We identify the upstream acetyltransferase actually is type 60, because the DOMA using budding yeast, fission yeast system basically demonstrate cell cycle is driven by phosphorylation. But apparently, type 60 here is has a function of amplification. So mechanistically, Aurora B activation require phosphorylation 232. And 232 thorium dephosphorated quickly by phosphatase. And acetylation here actually will increase Aurora B activity at the centromere to enable accurate chromosome segregation. And later on, we actually using transgenic mice, and we by using logging the TIP60 activated the form, which is S90D. Then we actually identify the spindle misorientation defect, and we identify actually TIP60 is acetyltransferase at the centromere during proto-metaphase transition. And then TIP60 activity is actually in the spindle cortex, which mediating astral microtubule association with cortex. So this basically is the diagram. So that actually indicating TIP60 has spatial temporal dependent effect. And later on, we identify another lower protein called Apollo-1. This is the POK adaptor and responsible for polokinase 1 localizing at the centromere and also dissociating from centromere upon the metaphase anaphase transition. And using zebrafish system, we actually identify surprise effect. Apollo-1 actually involving hematopoietic stem cell proliferation, but not biogenesis. And Apollo-1 is one of those protein contain low complexity domain and phase transition, actually hoping its interaction with polo kinase 1. So over the last roughly 2 decades, we actually identify several new centromere proteins. The long story short, those are human specific, and also the other groups that identify certain proteins such as BABA1, SMPEF. They also are all present in the mitochondrial system. So this suggests the mesosome system actually evolve a more complicated the centromere machinery for multicellular synergy, because some of those proteins localized on the centromere during mitotic cell and became membrane associated in the interface. And also later on, our collaborative study with Abby also showing those condensation-driven actually crossover in the meiosis as well. So the biomolecular condensation actually play a quite important role in the organizing the cellular structure and dynamics. The next example actually I would like to share basically is looking into the EB1. And this is basically its APC binder, promoting microtubule, actually polymerization. So EB1 is a droplet, basically in the, this is a lattice light sheet imaging. And we show basically EB1 through oligomerization became those droplet. And this actually, the liquid droplet actually study tracing back to the E.B. Wilson more than 126 years ago. And then later on, I think Tony Hyman and Clifford rediscovered this membranous organelle, and actually they characterize those molecular grammar for those intrinsically disordered region, and also Michael Rosen using the synthetic biology approach identify this multivariant interaction for organizing those biomolecular condensation. So we basically pinpoint the amino acids in the IDR region, and also we replace the IDR of EB1 with other protein has characterized IDR. So long story short, basically EB1 IDR has its unique function. So basically we created a functional separation domain, which can using other IDR to replace EB1 IDR, and also pinpoint the 6 amino acid. And basically EB1 biocondensation involving recruiting other XIP motif containing protein and serve as a molecular glue to basically to enable chromosome attachment to the spindle microtubule process. And we use EB1 condenser as a sponge to actually absorb protein from mitotic cell. One of the protein actually come up with SAMBR, and we also show EB1 phase separation is important for chromosome oscillation, basically between prometaphase and metaphase alignment. So here basically is the cryo-EM structure we solved with a collaborator of Jianye Zhang in our university. And we show actually EB1 by recruiting SAMBR and form those co-condenser and mediating chromosome oscillation during mitosis. And most recently, actually, we identify phase separation driven Aurora B CPC complex with WDR5, which is basically showing phase separation involving compartmentalization on chromosome. And we are actually in the stage basically taking the cellular dynamics approach and to study phase transition driven intracellular dynamics to intercellular dynamics. So in summary, we identify centromere machinery in human actually is involved, and multicellularity actually has extra function compared to the single eukaryotic system. And we think for these organoids is a powerful system to really study organelle dynamics and also multicellular synergy and some new and emerging function in organ physiology. And those are the collaborators and people contributed to the work I presented here. And I think this is a short talk, and as John indicated, we have a section in annual meeting, which is December 8th in the afternoon, and some of those people will be presenting in the meeting. And for those of you who could not make it into the Philadelphia meeting, and I think we have next April meeting with Chinese Society for Cell Biology with ASCB and also IFCB, APOCB. I welcome you to our meeting in China. Thank you all.
Thank you, Xuebiao. It's very
I'd be
good.
happy to take any question.
Yeah, very interesting. Are there questions? I can't see questions. Dinghuan, do you have a question? No. I do, Xuebiao.
Okay. Hey, John.
It's a pretty general question,
Yes.
but are centromeres themselves, are they molecularly the same in different cell types? And do they age, or are there changes related to their stability or SEMP-A maintenance during senescence or aging? Is there any?
I think that this is a really wonderful question. Actually, this can be couple of folds. We are actually look at the chromosome specific, meaning quantitative machinery of centromere, and our answer is different chromosome perhaps have slightly different geometry of centromere machinery. And I think that they should be cell type context dependent centromere, and in terms of the renewal, the turnover and assembling, we do think that there should be some differences, but that at this moment we are quite far away from making any conclusions. The specificity exists, but that's actually our job to do in the next few years. Thank you.
So how do you see or measure potentially these different centromere characteristics or protein
That's a good question. Yeah.
on different chromosomes in the same cell.
Yeah. I think we are in the process to there are ways to label chromosomes because they are cytogenetics. You can really look at the bending, but for us, we need to isolate the chromosome either using FACS sorter,
Yeah.
and then we will collect a sufficient amount of different number of chromosome, and then do the nanopore mass spec and try to identify the protein, the total protein on different chromosomes, and then we perhaps can get the quantitatively the centromere copies so that we can reconstitute back. And of course, the difficulties for certain cell type is the synchronization. So hopefully, by using this approach, try to get the idea of different the centromere on the different chromosome composition, we can use either expansion microscope or cryo ET to reconstitute, see the fine structure in centromere. Yeah.
So if you don't mind, one last question. This is a bit broader, but
Yes.
what do you think is the most persistent misconception we have about centromere identity or function?
That's a very, very good question. I think the textbook view is
Yeah.
it's a histone 3.3 variant, which called sample A. Sample A organizes this centromere nucleosome, and this is the building block for building the centromere. Of course, the new evidence showing some of the centromere protein has direct DNA binding. So the new model likely is sample nucleosome involving loading of centromere protein, and once those protein gets to the centromere, perhaps lock onto the centromeric DNA. So there are a couple of groups still working on the fine details. Yeah.
And somehow that's catalyzed by these protein condensates.
Yes, certainly the CCAN is assembled, driven by condensation. Yes.
Very cool. I don't know how to, I guess there may be other questions. Yurong, are you on the call? Do we see other questions in?
Yes, I'm on the call. No, I did not see people posting Q&A yet.
Okay,
Just
great.
conscious of time, I think we can continue. In the end, if additional time, we can take additional question.
That's great.
Yeah.
Right.
Great.
Thank you.
Thank you.
So now we get to have the pleasure of having these 5-minute lightning talks from early career awardees. This award recognizes first authors, mostly postdocs and graduate students, whose MBoC papers were selected by the editorial board as particularly meritorious, creative, or exciting. And it's a real pleasure to have these more junior folks here to give their perspectives on their own work. These brief presentations are always a highlight for me. They showcase the innovation and talent driving our field forward, and that's what we're hoping to recognize through these presentations and through these awards. After 3 presentations, we'll have time for questions. So please, if you have questions, put them in the chat and we'll address those questions in after 3 of the presentations. So first of all, let me see who we're introducing first. Our first presenter is Cátia Carvalho, and she is presenting on behalf of her partner, Matilde, and they are presenting, they're coming from Portugal, and they're speaking on a recent PhD work from Cátia's work. And I think Matilde Moreira is there. And Matilde, you're just gonna, or Matilde, you're gonna sit back and watch Cátia present part of your work. Is that correct?
No, it's actually the opposite. I'll be
It's the other way around.
Well, it's extremely early here for me.
It's okay.
So I'm glad
I'm going to sit and watch. Go ahead,
You
Matilde.
can watch, Cátia. Thank you. Matilde, go ahead. Thank you. Thanks for correcting me.
So, hi everyone, my name is Matilde, and today I will present you our work on the dual role of Zyke12 and its regulation by alternative splicing. Dynein is the major microtubule -N directed motor, and it is crucial and responsible for numerous processes inside the cells. The uniqueness of this motor contrasts with the vast diversity of cargos it transports, and so it needs adapter proteins to be recruited to those many different cargos. Adapters are dimeric coil proteins that bind dynein in the N-terminal and the cargo in the C-terminal. So how is one motor recruited to many different cargos? There's two ways that can happen. The first is to expand the adapter families and create new adapters for different cargo. One example of that is the hook family of adapters that in human cells, different paralogs, hook 1, 2, and 3, form different complexes that recruit dynein to different cargo. For example, the FHF complex is composed of a hook dimer and a copy each of the FTS and FIP1 proteins that by binding to RAP5 recruits dynein to early endosomes. The second way to recruit dynein to many different cargo is to have one adapter with different cargo binding domains. And today I hope to convince you that Zyke12, which is a C. elegans hook protein, is able to do just that. This protein in the C-terminal has a flexible linker, a transmembrane domain, and a cache motif that in the nuclear envelope are able to bind SUN1 and form the link complex. So as expected, we were able to observe Zyke12 in the nuclear envelope of the embryos of C. elegans, but surprisingly, when we looked at the epidermis of the larval stage, we saw that Zyke12 localized to early endosomes and not the nuclear envelope. So how is this tissue-specific localization regulated? We first looked at FIP1, which is part of the FHF complex, and saw that its knockout leads to the disappearance of Zyke12 from the early endosomes in the epidermis, while still localizing normally to the nuclear envelope in the early embryos, which means that FIP1 is required for the recruitment of Zyke12 to early endosomes, but is dispensable for its function in the nuclear envelope. We obtained the same phenotype when we deleted the part of Zyke12 responsible for interacting with the FHF proteins, but leaving the rest of the protein intact. And this further shows that the recruitment of Zyke12 to the different organelles, early endosomes, and nuclear envelope is mediated by different C-terminal domains that form different complexes. And we believe that the regulation of this dual recruitment is achieved by alternative splicing. Zyke12 exists in 3 isoforms: the full-length isoform B, a short isoform A that lacks the linker, transmembrane domain, and cache motif, and is thus unable to bind SUN1 in the nuclear envelope, and the long isoform C that lacks the flexible linker. The localization of Zyke12 in the nuclear envelope was not affected when we removed isoform A and B, which means that isoform C is sufficient for Zyke12's function in the nuclear envelope. And contrarily, the recruitment of Zyke12 to the early endosomes is reduced when we only have isoforms B and C, and even more so reduced when we remove isoform B, which suggests that isoform C is unable to recruit Zyke12 to the early endosomes. And we think that is because it lacks this flexible linker and is sterically unable to bind the FHF complex. To conclude, we have shown for the first time alternative splicing as a way to make an adapter multifunctional and multivalent in recruiting dynein to 2 different organelles, early endosomes, and the nuclear envelope. I want to finish by acknowledging Cátia Carvalho, which is the co-author of this work with me, Het Hogasman, who is our supervisor, and everyone else who contributed to this work, and also thank the MBoC and ASCB editorial boards for recognizing our work, and thank you all for listening today.
Great, thank you, Matilde. I think we'll move on to Mike Bischoff, who's presenting his work from University of North Carolina at Chapel Hill. Mike also, I don't see him on right now. There he is.
I'm there.
There he is. Good to see you. Mike is also starting his new group at in Munster, Germany. So he's very busy, and he's speaking to us from Germany now. Correct, Mike?
Yes, that's correct. Yeah.
Okay. Take it away.
Okay, I hope you can see my screen. Yeah, so first of all, I want to thank ACB and the MBoC editorial board for supporting me and the wonderful prize and publishing the paper and organizing this meeting. I just started a group in Münster, but this work is about my postdoc work in Mark Pfeiffer's lab. And I'm interested in mesenchymal cells. And recently, there's more and more findings that mesenchymal layers are quite crucial for organ shape. And I think they're quite different from epithelial cells, and therefore you have also very different mechanisms in morphogenesis. And there's another cool thing about cells like that are more on the mesenchymal side of the spectrum, and that is that they can determine patterns by themselves by harnessing different rules in different contexts. Cells can dynamically rearrange and self-assemble into patterns without external guidance. And if you want to know more about this, just this month or last month, we published a perspective on this. But how is this harnessed or used during organ sculpting? I think the answer to that is, to this moment, not really known. And the problem is, to understand such dynamic processes, we have to watch them as they happen. And learning them from like fixed tissue is a little bit like learning the rules of basketball by looking at a handful of screenshots, which is, of course, much easier if you have a video. And therefore, I established the Drosophila testis model of mesenchymal sculpting. So the Drosophila testis gets covered by these muscle cells that migrate onto it and then also sculpt it into its shape. Let me quickly walk you through the process. So first, the testis doesn't have muscles and is not connected to the genital tract. And then the two structures coalesce, which allows the muscles to pass over, to populate the entire structure. And then muscles start to elongate. And this actually sculpts the testis into this spiral shape. And you can see here that we can directly link migration defects and defects in their collective dynamics to shape defects. For example, if migration is slower, we see that we have fewer revolutions and like this club-like tip. And I think this is actually the perfect system to find regulators of this mesenchymal sculpting process. And therefore, we performed RNA-seq. We first FACS sorted the muscle cells, which worked quite well, and then we did transcriptomic. We compared different stages and looked at genes overexpressed in both stages. And we also looked within specific stages for genes that are highly expressed within their families. And based on this, we curated 131 genes together with Corbin Jones. And after that, we performed a large screen for adult defects by tissue, specifically knocking out these genes. And this screen is quite important for my career. So in the next slides, I just want to show you how it affected me. And all of this was actually done by me and a team of postdocs and interns. So it was a huge group effort. So next up after the screen, there was right away a follow-up paper about one of the candidates of the screen, which was plexin A, and which was important in the dynamic spreading of the cells during the migration, which is important for their sculpting. And in the future, I'm going to look at how actually the spiral shape is acquired, which is due to a non-conventional myosin that has introduced chirality to the system. And if I knock it down in muscles, it actually flips the chirality of testis. And here I'm also going to look at a candidate of the screen, which are Wnt4 and Frizzle2, which perturb pneumatic alignment. And it seems that the spiral formation is actually a dynamic rearrangement process of muscles that is happening. And all of this I am going to look at now in live, using my live testis system. So yeah, with that, I want to acknowledge everyone who worked on the project, and I want to thank you for your attention. And I want to bring to your attention that one of my postdocs, Sarah, is looking for grad school opportunities.
Great. Thanks, Mike. And that's a lot for you to do. That's a good career start. That's great. I made an error, which is not uncommon. It's still only 6:30 a.m. here. I was the first speaker in this session was supposed to be Dinghuan Deng, and Dinghuan is a physicist from Janelia Farms at the Hughes Medical Institute. So our third speaker in this little series session will be Dinghuan.
Hello. Yeah, a little bit time waiting, but finally it's my turn. Yeah. Okay. So today I would like to produce some of our recent works or working on the lysosome profiling. So my name is Dinghuan Deng. So I'm working at the General Research Chamber at HMI. So this work is basically working together with the, let me see. Together with the colleague from the Baylor College of Medicine and the HHM at Genelia. So since our lab is mainly focused on aging and metabolism, so we have developed a series of tools looking at the lysosome. So however, measuring the lysosome pH precisely remains a challenge. Traditional methods are using the fluorescence intensity or ratio-based with the two excitation ratios. It suffers from the proper concentration and laser intensity interference. Also, is time consuming. Here, we present a new technique that is using the fluorescence lifetime as an index by looking at the pH, because this technique is independent of the dye concentration and excitation intensity. Also, it's pretty fast. As 5 minutes labeling is enough for looking at the pH. So also, luckily, we found a lysosome sensor gene that is pH is sensitive to the lifetime. So basically, by looking at this calibration curve, you can see that the lifetime responds to the pH from linearly from pH 5 to pH 7. By looking at this calibration curve, we can convert the lifetime to pH easily. So we, to make the calibration more accurate, we also did the in vitro calibration, and the pH response showing the same, almost similar to the one with the in vitro. Also, we compare the film method with the traditional ratio method and found that the lifetime method has a high sensitivity. And also it can be used to analyze the individual lysosome pH. Here we use chloroquine as a method to control the pH in the U2OS cells. You can see that the film method shows a quite uniform results compared to the ratiometric method. So it's more sensitive and robust. By using this method, we have successfully applied it to the mammalian cells such as astrocytes. It has a narrow pH range in the control cell. When we treat it with the soylent starvation, we see that the pH increases the heterogeneity from the control. For microglia cells, we treat it with the soylent starvation and also LPS activation. You can see that for the certain starvation, the cell becomes less acidic. But for the LPS activation, we do see the cells becomes more acidic. We did this purposely to verify our system. So for the neuron cells, when the cells become mature, the cell definitely will become more acidic. So most of the lysosome goes down to below to pH 5. So this is almost the detection limit for this lysosome sensor. We also trying to apply this method to the live worms. So this is the main model we are researching in our lab. So since we are trying to look at the different organelles, we found that the hypodermis is reliable for the pH measurement. But when we are really want to trying to look at the intestine, the autofluorescence will make the interpretation more complicated because the autofluorescence is really strong in the intestine. So in this case, we are trying to developing a two-component analysis for need to use to dissect the signal, also separate the autofluorescence. So with this, I would like to summarize our results. So basically, it enables fast and quantitative pH profiling even for the individual lysosomes. This reveals the pH heterogeneity and dynamics across the different cell types and conditions. We try the mammalian cell and see elegance, but definitely you can try the model. And I think this is a powerful tool for studying the lysosome biology. Yeah. Finally, I would like to thank our collaborators and all the support teams. Thank you very much.
Thanks, Dinghuan.
Let me stop the skin selling.
Oh, I'm just looking at the titles, I believe I'm looking at. So I have a question for you, Dinghuan.
Yes.
So pH is one thing to measure in lysosomes. Should we be measuring calcium flux, membrane potential, other things in lysosomes as well? And do you imagine multi-parameter readouts with some of these sensors in the future?
Actually, we are developing a bunch of tools, not only the calcium. We are also doing like the EM imaging, cryo-EM imaging, also like other tools for looking at the lysosome, because this is one of the tools we are trying to developing. So we do have a bunch of tools, but looking at those features.
Can you see them fuse? Lysosomes fuse with the.
Oh, you mean the contactor?
Yeah.
We do have someone working on this project, but we are trying to use the cryo-EM and also the fluorescence to look at those
That's very cool.
Yeah.
That's very cool. Thank you. I don't see any questions in the chat, so I'm going to continue my my little tour of questions, and I'll ask Mike. So you've got, what did you say, 131 transcripts you identified that were highly expressed in stage-specific gene expression? Is that correct?
Yeah, this is everything taken together. So we basically made a list of candidates based on the RNA-seq, and then did this like candidate gene approach of knocking them down.
And then, so now with 131, now you've got, and how do you prioritize them? So you've got 29 that you've done knockouts of now. Is that correct?
Yes. Yeah.
How do you prioritize them? Is it just based on expression, or are there other criteria that you're using to prioritize which ones you're following up? And what surprises have you got so far?
Okay. So what I now follow up is, I would say, I picked the low-hanging fruit, obviously, like in terms of knockdown phenotype. And what I was looking for most is, I mean, it's pretty clear that when they're not migrating, that they cannot sculpt the tissue. but for those that basically covered everything, but yet were not able to sculpt the tissue. And there, I think the most interesting ones are Wnt4 and Frizzle2, which perturb the nematic alignment. And then you see that like instead of a nicely shaped spiral, you basically get a weird sausage shape. So these are obviously quite interesting. And then there is this adhesion G protein coupled receptor, latrophalin, which might act as a mechanosensor and also leads to these nematic defects. So this is also one I'm going to follow up in the future.
There's some pretty cool phenotypes for you to be looking through. So you're doing knockdowns, and then are you tagging each one of the proteins that you're interested in as well, and looking at its function and localization within the cell?
Yeah, sure. I mean, there is a wide array of flag resources there, and we can also quickly make our own constructs here. But yeah, right now I'm focusing on certain regulators and following them up.
Great. And then my question for Matilde and Cátia. So your title says that ZIG12's regulated by alternative splicing, right? Its function is regulated by, or its adaptor function is regulated by alternative splicing. But do you know whether the splicing itself is also regulated? Is there evidence of that?
We actually don't know how the isoforms are chosen per se. It may be because of the availability of the binding partner. So FIP1, which is part of the complex that binds to the early endosomes, is not present in the embryos, for example, and so their ZIG12 is only function is in the nuclear envelope, but we're not sure yet if that is the case.
Great, thank you. I do not see you wrong. Do you see any questions in the chat that I can't see?
No, I do not. Just
A quick
seeing
question.
Go ahead.
Can non-discussion
Oh, yes, of course. Go ahead.
write in the chat? No, I'm not sure if they can.
I think the people can write to the Q&A. So everybody, please write your question in Q&A. Yeah.
Okay. We can always come back to questions, but I'd like to move on. I think we should move on to the next round of lightning talks. So the first speaker is Z. Craig, and Z, haven't met you yet. Welcome. And are you still at the University of Michigan?
I am, yes.
Maybe that's great. So I think we could probably get through the four speakers in this session. So we'll move through these in that order. Z, go ahead.
Can you all see my screen and hear me okay?
Yep.
Okay, so hi everyone, my name is Z. Craig. I'm a postdoc at the University of Michigan, and today I'll be telling you about our work investigating how endolin tunes contractility and regulates barrier function during rho flare-mediated tight junction remodeling. So the focus of this paper is epithelial cells, which you see here in this image, where we're looking at the developing frog epithelium. I'm sure this audience is familiar with epithelial cells. These are the cells that line and compartmentalize our organs and give them specialized function, one of the most important being barrier function. However, these cells are very dynamic and they often go through cell shape changes. So a question that we can ask is how are cells able to maintain their barrier function during cell shape change events? One way that I'll be telling you about today is through the rho flare repair pathway, which I'll show you in this video, where tight junctions are marked by the protein ZO1 shown in magenta, and there's also a probe for the active form of the small GTPase rho A shown in green. And as this movie plays, you can follow along with these steps in the schematic on the right. As junctions elongate during cell shape changes, tight junctions can break, causing a leak. This causes an influx of calcium and the recruitment of the P115 Rho GEF, which activates Rho, driving a downstream actin-myosin contraction. The junction then shortens, and the tight junction is repaired and reinforced over baseline. Several previous papers in the lab really looked at the upstream mechanism of this event. However, for this paper, we were really interested in the downstream events. As I'm sure you've noticed from the video, Rho flares are very transient, which suggests that contractility is tightly regulated during this process. So we chose to look at the anilin scaffolding protein due to the fact that anilin has been shown to both positively regulate Rho and negatively regulate Rho, and has also been shown to regulate cell-cell junctions, and we found that it localized to flares. So what we found is that when we knocked anilin down and tracked the intensity of active Rho over the lifetime of the flare versus controls shown in light blue, anilin knockdown embryos shown in dark blue, had these more intense Rho flares, which suggested that anilin negatively regulates Rho here. But we also found that these Rho flares decayed faster, suggesting negative or positive regulation of Rho. We also found that there was an effect on Z01 following the Rho flare, where Z01 intensity was only reinstated in anilin knockdown versus reinforced in control embryos. And we wondered if the changes in dynamics were creating insufficient contractility, which was leading to the reinstatement instead of reinforcement. So to investigate this, we looked at actin and myosin at individual Rho flares, and we found that actin intensity very closely mirrored what we saw in Rho intensity and was more intense. However, more interestingly, we found that myosin intensity was nearly abolished in anilin knockdown embryos at flares, shown in the far right panel. We found this to be true over multiple examples and showed that the maximum intensity of myosin is significantly decreased in anilin knockdown embryos at flares, which suggests that the time that Rho is active is more important in sustaining myosin contractility. And to kind of recap this and bring you back to the schematic that I showed you in the beginning, the changes in Rho flare dynamics created insufficient contractility. And what this meant was that these junctions were actually unable to shorten adequately, which we measured as a change in junction length. However, you may have noticed that I've stated that these junctions are reinstated, they're just not reinforced, meaning they are repaired. So what is the consequence, if any, to the organism? To answer that question, I'll show you one more video where we have a control embryo on the left, anilin knockdown embryo on the right, and what we're viewing is a fluorescent molecule called flosin-3, which allows us to see barrier leaks as a change in flosin intensity at the junctions. Here, this is being shown in a fire lookup table, so low intensity is shown in blues or purples, up to high intensity shown in yellow or white. And as this movie plays, what I think you'll be able to appreciate immediately is that anilin knockdown embryos have an increase in flosin at their junctions at baseline, but you'll also notice that there are more of these leaks and they are more complex or multicellular, meaning that multiple cells are being implicated in these breaks at once. And so together, this data shows that anilin is required to tune contractility as well as to regulate barrier function during tight junction remodeling. With that, I would just like to thank the organizers at MBoC for recognizing our paper and allowing me the chance to talk about our research, the Miller Lab, as well as our funding resources. And I have since left Anne's lab for my PhD and moved to Rachel Siemens' lab for my postdoc.
Thank you very much. Very cool. Vasilios Raphael from Edinburgh, originally from Greece.
Yes, that's me. Can you all see my screen?
We can.
Lovely. So I'm Vasilis, and today I'm going to talk to you about some of the work that we did for our 2024 publication that is about how the outer kinetochore components, KNL1 and NDC80, can regulate axon and cell body positioning and C elegans. So kinetochores are multi-protein structures that are composed of more than 100 different types of proteins. The inner kinetochore associates with DNA, whereas the outer kinetochore with microtubules, mediating this way chromosome segregation during cell division. So within the outer kinetochore, there is a KMN network that consists of KNL1, Mis12 complex, and NDC80 complex, through which the microtubule attachments are being formed. So this acts as a signaling device that coordinates the mechanical coupling of microtubules to the progression of the cell cycle. So over the last few years, work from different labs has shown that kinetochores are implicated in several aspects of neuronal development, such as dendrite formation and regeneration, synapse formation, and what we showed in this paper, brain and axon development. So I first started working on this paper in the beginning of my PhD, following up on previous findings from my supervisor, where they showed that several kinetochore components localizing outside of the nucleus in neuronal processes, as you can see here in C. elegans embryos. So then we employed a GFP-based degradation system with which we can degrade these essential proteins post-mitotically in head neurons in C. elegans. And what we saw was a disorganization of the C. elegans mini brain, which is termed as the nerve ring, and this is composed of more than 180 axons, and also a complete dispersion of the cell bodies of the head sensory neurons, as you can see here. And in data that I'm not showing here at the moment, we saw that on a single axon level, depletion of kinetochore components led to axon misguidance. So then, trying to understand where this phenotype comes from, we employed a gene replacement system in which we degrade the kinetochore components KNL1 and NDC80, and at the same time we express mutated versions of these proteins in the background of the respective degraders. So we mutated the signaling module of KNL1 and the microtubule binding domains of NDC80. And what we saw, as you can see here in the graph, that the dispersion of the ganglia of the head neurons is significantly increased compared to the control, suggesting that both the signaling and the microtubule binding interfaces of kinetochores are important for the neuronal functions. And then to get to the source of the kinetochore's role in axon development, we started looking at early events of brain formation, specifically in the embryo. There, a group of pioneer neurons extend their axons that form this tight bundle, following a ring-like trajectory, which in the future, which is the framework for the formation of the future nerve ring. So we developed a system where we could visualize all of the pioneer neurons with green, and with magenta, we could visualize just a single pair of pioneer neurons, the SIAV, so that we could track down the trajectory of single axons within the axonal bundle. So we focused on the microtubule binding mutant of NDC80 that I mentioned earlier due to its importance in neuronal function. And here on the left, you can see in the wild type, the axons of the pioneer neurons extending normally, forming the tight bundle and forming the framework for the nerve ring. Whereas in the microtubule binding mutant, we saw several defects. Most importantly, the SIAV axon fails to extend and incorporate in the nerve ring, and also axons of other pioneer neurons seem to also fail to incorporate in the axonal bundle. So this suggests that kinetochores are needed during these early events of axon development to facilitate proper guidance and bundling that is needed for nerve ring formation. So the takeaway message from today's talk is that in this publication we saw that kinetochores are repurposed from cell division, from chromosome segregation to brain development by utilizing both the signaling and the microtubule binding interfaces to facilitate nerve ring organization by regulating axon placement and bundling in these early events of embryogenesis. And at this point, I would like to thank you all for your attention, and also everyone, every single member of the Chirambathu Lab at the University of Edinburgh, and members and people from the lab that are not in the lab anymore, whose work was very significant for this paper.
Thank you. Our next speaker is Amy Strom from Princeton. Nice dog, Amy.
Oh, 1 second. I can't hear you, Amy.
Yeah, sorry, you're muted.
How about now?
Good.
Okay, yes, sorry. I was telling you that I'm showing you this picture of my dog for scientific reasons. He's at the beach, and I am including this because I've been thinking a lot about intracellular interfaces. So this is my land-sea interface to set the scene. So unstructured regions of proteins are also known as intrinsically disordered, or IDRs, and they lack a fixed structure. And interestingly, proteins involved in transcription and chromatin have the highest fraction of disorder, whereas proteins involved in metabolism and homeostasis are the least disordered. That means when you put them into AlphaFold, you get things that look like down here. Transcription factors have small folded domains, but really the rest is just a big noodle. And what do these kind of disordered regions of proteins do? We just heard a great talk from our keynote speaker about biomolecular condensation. So these transient and flexible interactions are also multivalent, which results in the formation of these membraneless organelles or biomolecular condensates. And specifically in transcriptional regulation, a lot of work's been done recently to define that IDRs from mediator complex interact with those from specific transcription factors, and they together form this complex that recruits RNA polymerase II to be loaded at transcriptional start sites. And the work I'll tell you about today is investigating the regulation of seeding this whole complex at specific chromatin sites. So why, if all the interactions that go into forming this condensate are kind of possible anywhere in the nucleus, we get on chromatin condensation that's transcriptionally productive and not off chromatin condensation that would be sequestering all this important machinery away from the sites where it needs to act. And to study this, we are looking into a protein called BRD4, which is a BET family protein, and it has one of these IDRs that mediates phase separation. And as a postdoc in the Dinghuan lab, we have this cool tool that makes light-inducible condensation out of these IDRs. So you can start by expressing it in a live cell, turning on the light to induce phase separation, turn off the light, it disperses again, and do this repeatedly. And when you do this with a construct that just contains the intrinsically disordered region, and then look at where those droplets form, like by overlaying pictures from the first cycle, second cycle, or second and third cycle, they don't overlap. They're randomly located. But if we do this with the full-length protein that has the bromodomains that bind to acetylated chromatin, we see that these condensates are forming again and again at the same nuclear sites. And we can even disrupt the chromatin interaction with a small molecule called JQ1 that fits in that acetylated chromatin binding pocket and now make that randomly localized again. So the interaction between BRD4 and chromatin is what's localizing the condensates to specific areas within the nucleus. And this project was a collaboration with simulation, coarse grain simulation collaborators who made this cool system where we have a chromatin polymer that has acetylated and non-acetylated tails along it, and then are representing the BRD4 with two spheres representing the N and C termini, and we can oligomerize those at different valences in order to measure what's called the critical valence with or without chromatin. And what we find is that with chromatin, the critical valence is 2. Without chromatin, it's 6. So that means that you only need to dimerize BRD4 when there's also this chromatin template around to form a condensate. Whereas without that chromatin template, you need essentially a hexamer of that BRD4 in order to initiate condensation. And we can also, in a slightly bigger simulation, ask how often do these condensates form on that chromatin or off chromatin? And what is the kinetics of that? How long does it take to form one on or off? And it turns out that it's both a switch-like process where at a variety of acetylation fractions, we get really only on chromatin nucleation when the chromatin is present, and entirely off chromatin nucleation when we mimic that plus JQ1 state that disrupts the interaction. The delay time of nucleation, the time before a condensate forms, is much increased in the off chromatin state. So these two types of simulations are telling us that that chromatin binding factor enhances both thermodynamic and kinetic aspects of BRD4 phase separation. So with this parallel experiment and simulation, we learned about this kind of interplay of the condensation and chromatin binding that underlies this targeting. It's actually competition within the nucleus where acetylated chromatin contributes both to that thermodynamics through valence enhancement and kinetics through seeded nucleation to trigger BRD4 condensation specifically at chromatin sites. However, perturbations to the system, like disrupting the chromatin binding or reducing acetylation levels or even overexpressing BRD4, can shift the balance toward off-chromatin BRD4 condensation, which is randomly localized and leads to aberrant transcriptional sequestration. We think this model of balancing on and off chromatin is generalizable to other chromatin binding and phase separating proteins, considering how to use this for quantitative knowledge to kind of rescue that balance in disease states like cancer. This project was completed while I was a postdoc at Princeton working with Cliff Brangwyn, and the simulations were done by Yuri in Will Jacobs' lab. I'm now at Genentech in Discovery Oncology. Here's my email if you want to follow me or go to amystrom.com. And if I have 10 more seconds, I wanted to show one cool bonus data because I was inspired by our keynote speaker. And this is just an image where I have that light-inducible formation of a condensate, and this is attached to two different chromatin loci within a living cell. And here I'm showing that the surface tension of that condensate can actually put force on those chromatin loci and move them towards each other in a living cell. So this is actually from a different paper, but I thought it was fun to think about the parallels between interphase chromatin loci movement and kinetic horror-based chromosome movement. Thank you.
Thank you, Amy. Okay, I have questions for you too, but we'll move on to Alicia, and then we'll have questions at the end. And please, as Yurong has mentioned, if you have questions, drop them into the Q&A section on your browser. Go ahead, Alicia.
All right, I'll be quick for the sake of time and to then hopefully have more time for questions. Okay, so the story I'll be telling today is focused on copper deficiency in a rare genetic disease called Menkes disease that is caused by the inability to absorb copper from the diet. This figure shows some of the characteristic features of Menkes disease, which includes atrophy of the cortex and cerebellum in the brain and the accumulation of large distended mitochondria in the cell bodies of Purkinje neurons. There's currently no effective treatment for Menkes disease, and we do not fully understand why copper deficiency is harmful to the brain. So the goal of my project is to discover fundamental mechanisms related to copper homeostasis that are required for human development, but also that are involved in more common diseases like Alzheimer's, which is also associated with brain-specific copper deficiency. The overarching strategy that my lab uses involves first creating mutant cell lines to model our disease of interest. We then use systems biology approaches to gather information from these cells to generate hypotheses that we test in cell and animal models of disease. As I don't have the time to review all of the data that we gathered from these systems today. I'll point you to the manuscript where the PubMed ID is at the bottom of the page. And I'll simply highlight for you that we observed increased activity of the mTOR kinase, as well as downstream protein synthesis. And we saw this in both cellular and animal models of copper deficiency. And this led us to further explore the functional consequences of this increase in protein synthesis and copper deficiency that I'll highlight for you today in a fly model. So here you can see a trace of two neurons. On the left is the control, and on the right is a copper deficient neuron that is overexpressing a protein called ATP7 and is constantly releasing copper from the cell. Overexpression of this ATP7 is a cell autonomous model of copper deficiency, and hopefully if you compare the dendritic arbors, you can see the dramatic defects here in the processes in this neuron. However, if we overexpress S6 kinase, which is downstream of mTOR and ultimately promotes protein synthesis, you can see that we partially rescue this dendritic arborization defect. Additionally, we can look at the mitochondria within the processes of these neurons. So you can see here, hopefully you can see it okay on your screen, mitochondria in green, indicated by white arrows. They're plentiful in the control neuron and almost completely absent in the copper deficient neuron. However, and if you quantify it here, you can see how dramatic these defects are. But if we overexpress S6 kinase, you can see we fully rescue this phenotype and restore mitochondria to these neuronal processes. So based on this, what I hope to have shown you in this talk is that genetic activation of protein synthesis pathways either fully or partially ameliorates these dendritic neuronal processes phenotypes in this cell autonomous model of copper deficiency in flies. And from this, we conclude that mTOR-dependent protein synthesis is a resilience mechanism in neuronal copper deficiency. I'd like to acknowledge our funding sources, as well as everyone in my lab and our collaborators who contributed to this work, including my cat as well. And I'm happy to take questions in the Q&A period. Thank you.
Thank you, Alicia. So if I could start with a question from you, or for you. Why mitochondria? Why are they deficient from these processes? And what about other organelles?
Certainly. So I think one of the reasons we think mitochondria are primarily affected is that you actually need copper for the proper assembly and function of the respiratory chain. And so it makes sense that if you're failing to assemble complex 4 and respiration is impaired, that your mitochondria are going to be really dramatically impacted. But we also have a lot of data to show that lysosomes are impacted, that the ER is impacted. So we think mitochondria are an important driver of this phenotype, but certainly other organelles are likely to be involved too.
And just remind me, what's the link between mTOR and mitochondria, S6 kinase?
So I think there's too much to say there in the context of this webinar. I think briefly, mTOR is an important regulator of autophagy and mitophagy. So there's certainly a component there, but I think we're seeing more in our systems biology approaches than simply mitophagy. And I think there's probably a kind of interplay, a crosstalk with energy metabolism of mTOR as a nutrient sensing pathway. And so the mitochondria is obviously really important for energy production. And so there's something there that's really interesting, because you wouldn't expect these nutrient deficient cells that can't really use their mitochondria to make ATP to be upregulating mTOR. So there is an interesting something or other happening there that we're interested in further exploring.
I'm wondering about ROS and ROS responses, and actually wondering about peroxisomes in these processes too. Any idea?
Let me see. Sorry, I did not realize the Zoom updated. I didn't realize that I was still sharing my screen. Would you mind repeating your question? It was peroxisomes?
I was just wondering, I was wondering about ROS, if you know anything about the state of reactive oxygen and whether peroxisomes are affected and maybe compensate for loss of mitochondria or vice versa.
Certainly. So I highly suspect that there is increased reactive oxygen species in these cells. We haven't measured it. That's something we like to do in the long term. And certainly if that's the case, it's possible that peroxisomes are involved. But I think that's a good pin in that we really need to do flow to see how these cells are potentially increasing ROS and what mechanisms might be responsible for that.
Thank you. Amy, I'm wondering about this. It's remarkable inducible light inducible condensate.
I know, I skipped over all the details.
How does it work briefly?
Briefly, it's based on oligomerization states. So we have a 24-mer sphere, which is the ferritin core. And onto each of those subunits, we have tagged 1/2 of a light-inducible heterodimer pair that we stole from plants. So it's an eyelid, so it's like a LOVE2 domain eyelid on that 24-mer sphere. And then the other half is tagged to your IDR of interest. And you express both of those, and they kind of diffuse themselves until you turn on the light, which induces that interaction between the eyelid and SSPB. And now you have like a 24-mer or up to 24-mer oligomerized valence of your IDR of interest. And that how much is it oligomerized is controlled by the ratio of expression levels between those two pieces. So in each cell, you might have a slightly different valence, and you can build that phase diagram from this variety of valence and ask at what valence do you see these condensates start to form.
So you found this valency, that the critical valency on chromosomes is 2 versus in the nucleoplasm, not on chromosomes, is 6, right? Is that actually, does the valency actually change on chromosomes, or is this just a critical level required to induce the condensation?
Yeah, we think that the valence in the endogenous case is coming from the chromatin, that like multiple nucleosomes next to one another that have acetylated regions will each bind 1 or 2 BRD4 proteins. And once those come together, that's enough to nucleate this condensate to grow at that site.
But when you induce condensates, like you showed in that last video, that how do you differentiate between cause and effect, right? Or whether you're actually inducing the condensate to form in an artificial way that would bring in that example chromosomes together versus chromosomes coming together and forming a condensate to help stabilize that chromosome condensation.
Yeah, so in the last video, I have both I have an additional piece. So I have that condensate that I formed with the light inducible droplet. And then I also have tagged pieces of the chromosome that have kind of sticky ends that will bind onto the surface of that. And I can, for example, hold the laser on and have that condensate stay large, and the chromosome loci will stay attached to the surface of that condensate and not move towards one another. But then when I actually turn off the light, which shrinks that droplet, that's what actually pulls them together, provides the force with which to pull those together. So I can compare keeping that light on, keeping the chromosome, keeping the condensate large, and that keeps them separated. And then when I turn off the light, it shrinks that condensate and brings those two loci together. So again, of course, this is an artificial system, but I do think that the formation and dissolving of these condensates rapidly over time might be a way of kind of stirring different regions of the chromosomes.
How long can you maintain that?
Mostly the cell moves out of the way of the laser before I dissolve the condensate, but minutes to tens of minutes.
Interesting. Well, maybe we'll talk a little later. I'm just wondering.
Yeah, for sure. Email.
Wondering about the application to other condensates. So maybe I'll follow up.
Yeah, thanks.
That was great. We have to be aware of the time. So there were other talks, and there were other talks for which I had questions. So I have a question for Vasilios. So on the signaling interface and the mitochondrial, or excuse me, the microtubule binding interface, and so how do they talk to each other during, for example, an error correction? Are they mechanically coupled, or I mean, how do you think they communicate with one another?
Yeah. So in mitosis, I'm suspecting you're asking, right? Because in
Right.
the neuronal context, we don't really know what exactly is happening yet. We haven't really figured out the mechanism by which these two interfaces, you say, they like the crosstalk between them two. But so in mitosis, basically the signaling part, the KNL1, which is a large protein of the outer of the KMN network, basically is like a scaffold protein, and the N-terminal of the protein, which is the signaling, recruits several components that regulate the spindle assembly checkpoint. So in when, for example, microtubules are not properly attached to the kinetochore, then the spindle assembly checkpoint is activated and monitors basically the proper attachment of microtubules so that cell cycle can progress. But yeah, like I said, we don't really know what exactly is happening in neurons exactly, because we don't really, like, these proteins are found in the cytoplasm, so they don't really associate with DNA in this particular instance. And the relation to microtubule also, the relationship to microtubules, it's not, like in the neuronal context, it's not established yet. There's a research that has shown, other papers that have shown that depletion of kinetochore components can increase the microtubule dynamics, but like in this paper that we published at MBoC, we didn't see any difference in microtubule organization. So yeah, there's a lot of open questions at the moment.
And so my question really, it sort of ties to Z's talk regarding these row flares, and I'll just connect to Z now. I'm wondering, so you've got this, I guess, danger response, right? There's some response to epithelial integrity, and I wonder what you think actually senses the injury. Is it like a molecular sensor, or is it is it altered tight junction? Is it altered tension? Or, which would be much more mechanical, which is what I was thinking about with Vasilis's talk. Is there a defined molecular sensor, or does the system self-detect through just loss of tension?
That's a great question. We have shown in a previous paper that the junction elongation serves as a mechanical signal. And so we know that a mechanosensitive channel is activated. However, the exact mechanosensitive channel hasn't yet been discovered or characterized. So that would be, like a change in tension, activating that channel and allowing calcium to open. However, I don't think it's outside the realm of possibility that there are multiple sensors in place to detect something that is so catastrophic as a leak between cells.
So do you think the flare can sort of activate before there's an absolute loss of tension or calcium loss?
That's a great question. We do see a range of row flare morphology as well. So in some cases, we do see that the flare is activated before there's a complete loss of Z01 or like a tight junction protein. In some cases, I think particularly if they're quite fast, if the cell shape change is very drastic and the tight junction proteins are kind of pulled apart very quickly, you get that complete loss. But it does seem that the row flare can be activated as the loss is happening and before there's a complete or like catastrophic break.
Great. Very cool. I think we'll wrap things up there. If there are there other questions? Our audience seems to be pretty quiet, but that's okay. You wrong?
Yeah, I did not see additional question from Q&A.
Okay, great. Well, I want to thank you all for coming and joining us today. I mean, it's great to see these, first of all, some of these great new technologies and where they're going, and younger faces in the audience, or younger faces speaking about their work. It's great to see the excitement about your work. I also want to emphasize, it's just amazing the breadth of work that's getting published in the journal, from the neuronal stuff that we saw to Dinghuan's stuff, all the Amy's. It's wonderful. I really like to see these big screens. That's my personal thinking. But Mike, your project is going to take off. You've got a lot to do, and I hope all those data are available for everyone to help you. There's a lot of questions there. So thank you all for joining us, and I want to remind you all of the ASCB conference that's upcoming, and in Philadelphia. The dates of that conference, Hurong? I can't remember off the top of my head.
December 5.
Great. In Philadelphia is an MBoC ASCB joint conference, Cell Biology 2026. So thank you all for joining us. Thank you to the MBoC team for organizing this. Thank you, Hurong. Thank you, Xuebiao, for a wonderful overall presentation, and we look forward to seeing you at ASCB as well.
Yes. See you later. Have a good day.
Okay, thank you all. Goodbye.
Thanks, everyone.